Greetings to all. Let's enter into the course of Introduction to Bioinformatics. I'm your course instructor, myself Dr. Elamathi Natarajan. Currently, I'm working as an assistant professor in the Department of Bioinformatics, Kalinga University. Now we get into the module 2, where we can see the different types of analysis, what we can achieve by the bioinformatics tools with this biological data. So first thing, very basic foremost thing is that we will go for the sequencing of any genome, either part of it through exome or else transcriptome, or else we can go for a whole genome sequence if applicable. The very basic criteria is that we should go for the assembly of those particular genome sequence. So this can be achieved by using different kind of computational tools and algorithms lies with them. For them, the very basic thing which is required for the bioinformatics application is that sequence analysis. So we'll get into some kind of detailed analysis of the basic sequence analysis. Yeah, so the genome annotation can be achieved in different way through like machine learning approaches, sequence-based approaches, or else through comparative genomic approaches, or through evolutionary phylogenetic approaches, it can be achieved. So, the basic sequence element can be achieved by using the pairwise sequence element approaches as well as the multiple sequence element approaches. Once it is done, then we can go for the comparative genomic approaches where we can utilize the phylogenetic analysis. It is one of the best scope in the bioinformatics, where here through doing the phylogenetic analysis, we not only come to know about the molecular evolution of a particular data, either a protein or gene, or else like we can also find out the how distinct the genes or proteins similar to the other taxonomic lineage, or else we can also see the where the particular gene or protein has been evolved and what could be the ancestor from where the modification happened, how it evolved. Such kind of notable changes can be studied via this phylogenetic analysis through molecular evolution study. So then we can do the next differential gene expression analysis. This can be achieved by using the basic conventional techniques and like RT-PCR or microarray, or else nowadays we recently focus on the next generation sequence analysis, where the gene expression is studied through differential expression as well as, and it is achieved via the computational approaches is mainly helpful, and diagnosis part is majorly act as a role in the gene expression study in case it is act as one of the diagnostic technique in case of cancer and also some of the genetic disorders. So we can also study the regulatory elements and also the functional part of those regulatory elements, and the specific metabolic pathway studies we can predict and also analyze them in the useful way, and also we can do some of the system biology approaches, and neural network based studies can also be achieved via this, not only in case of disease but also in case of biomarker discovery in the finding of novel targets and as well as novel ligand. We can go for the, in case if the experimental techniques of protein structure is not available, then we can also go for the molecular theoretical protein structure prediction via the molecular modeling approach, and as well as the docking of the protein and ligand compound is also achieved through the molecular docking approaches via the computational tools is also possible over there for the better biomarker discovery and also the novel drug discovery. We can use this. So like the evolution period, you know, that starts from the DNA, and now we are into the not only in the Human Genome Project, but now we have achieved more along with that for the genome of many vectors, as well as the significant species and significant model organism genome is there. Now we are in the stage of reaching the personalized based medicine. We are approaching there, and it is soon away reaches everyone's market to get a better personalized medicine based diseases, to solve the diseases in a better way. So we can also perform the different structural annotation part as well as functional annotation part. Here we concentrate on the regulatory parts of the either the nucleotide sequence as well as the protein sequence, where we concentrate on the structural part as well as the functional site annotation is also can be carried out here. So we can also identify the new genes and validate the new genes via the comparative genomic approaches through the regulatory functional sites. So by using these annotation and differential expression studies, where we can understand the function of genes and also the other parts of the genome and their system in a better way. So here, apart from the function, we can also study the structural part of the genome as well as the protein part. Thank you.